Today topic is Staphylococcus. Staphylococcus is small spherical gram-positive cocci arranged in grape-like clusters. It has two variants. Mainly the first one is Staphylococcus aureus pyogenes. Pyo means nothing but pus. So these are pus forming, contains an enzyme coagulase which is coagulase positive, produces golden brown colonies, forms a toxin. Second variant is Staphylococcus albus or epidermis. It is commensal variety present in the skin and intestinal flora. It contains coagulase negative, forms white colored colonies, does not produce toxins. Third is Staphylococcus citrus, produces the lemon yellow colonies. Before going into details of the Staphylococcus aureus, we have to remember the main points. They are morphology, cultural characteristics, biochemical reactions, resistance, toxin, pathogenesis, lab diagnosis. First we will go with morphology. Morphology of Staphylococcus aureus. It is a spherical cocci, gram-positive, diameter of 1 micron, arranged characteristically in grape-like clusters, found singly or pairs. It is non-motile, non-sporing, non-capsulated. Sometimes capsule may be demonstrated. These are the pictures of the Staphylococcus aureus under the microscope. Cultural characteristics. They grow readily on the ordinary media. Temperature should be of around 10 to 42 degrees. These are aerobes and facultative anaerobes. On nutrient agar, after 24 hours, colonies are large, circular, convex, smooth, shiny, opaque, and raised. Most strains produce golden yellow pigment. Some may produce white, orange, or yellow, but mainly golden yellow pigment is seen. Produces oil paint appearance. It is the main character in the nutrient agar. On blood agar, they produce beta type of hemolysins. These are the golden yellow pigment on nutrient agar. And this is the beta hemolysis which is seen on the blood agar. On McConkies media, they produce pink colonies since they are lactose fermenters. In the McConkies media, the bacteria which is the lactose fermenters, they will produce the pink colonies. So Staphylococcus aureus is an lactose fermenter. Selective media like milk agar, salt broth, and Dudlam's media. Biochemical reactions. They ferment a number of sugars producing acid but not gas. They ferment a particular sugar called as mannitol. We should remember the name mannitol, which is not fermented by Staphylococcus epidermis. This is the difference, one of the difference between the Staphylococcus aureus and Staphylococcus epidermis. They reduce nitrates to nitrites. It is a lipolytic action on egg media. Resistance. Most resistant non-sporing bacteria. They survive on dried pus up to 2 to 3 months. They can withstand 60 degrees for 30 minutes. Toxins. It produces a number of exotoxins. They are broadly divided into cytolytic toxins, enterotoxin, exfoliative toxin. In cytolytic toxin, we can see subtypes. They are hemolysins and leukocidins. We will go with cytolytic toxins. Cytolytic toxins, the first one is hemolysins. 4 antigenically distinct hemolysins are produced. They are alpha, beta, gamma, delta. Alpha toxin, most important in the pathogenicity. It is a protein inactivated at 60 degrees. The toxin is denatured above 100 degrees. It is leukocidal, cytotoxic, and demonocrotic property. It can be toxicated and it is antigenic in nature. Beta toxin, usually seen in animals. It is hemolytic for sheep, characteristics of strains of bovine origin. Bovine origin is nothing but it's an animal origin. Bovine means animal origin. Gamma toxins, weakest of the all toxins. And delta toxins are human variety. It is lytic to human beings. It found in strains from the human lesions. Beta toxins are the animal variety. Delta toxins are the human variety. And second variant in the cytotoxic toxins are leukocidins. The toxin is composed of two fractions which are S and F. If both S and F are together, then it is toxic. Otherwise, no. Enterotoxin are responsible for the manifestation of staphylococcal food poisoning. Like the characteristic, we can see nausea, vomiting, diarrhea occurring within the 6 hours of consuming contaminated food which contains the Staphylococcus aureus. Heat table, there are 6 soluble toxins A to F. Mainly the enterotoxin F is reported to be responsible for the TSS. TSS is nothing but toxic shock syndrome. Epidermolytic toxin or exfoliative toxin. It leads to a variety of exfoliative skin diseases or disorders like Ritter's disease, localized bulbous impetigo, and SSS seen in the children below 4 years of age. SSS is nothing but Staphylococcus scalped skin syndrome. Like these, we can see the pictures of Ritter's disease, bullous impetigo, and SSS in the further video. Pathogenicity. It causes a majority of acute pyogenic lesions in man. These lesions are characteristically localized in contrast to spreading lesions of strepto. Lesions produced can be mainly four types. They are cutaneous lesion, deep infection, acute toxemias including food poisoning, exfoliative disease, toxic shock syndrome. We have to remember the pathogenicity. It is the important feature in every bacteria. And we will go with cutaneous lesions. They include boils, styes, furuncles, folliculitis, abscesses, carbuncles, pemphigus, impetigo. Staphylococcus also frequently causes sepsis in the wounds and burns. Majority of the hospital cross infections are of staphylococcal origin. Cross infections, like we had already discussed in the previous video of infection, what are the cross infection, definition is also given in the previous video. This is the styes in the eye. This is the first figure shows the pemphigus and the second figure shows the furuncles. Deep lesions, commonest infection in the acute osteomyelitis. 90% is by the staphylococcus, and in the respiration tract it causes tonsillitis, pharyngitis, sinusitis, and pneumonia. Pulmonary abscess, breast abscess in lactating mother is an important cause of streptococcal infection. Meningitis, endocarditis, pyelonephritis, and renal abscess also cause. It may also cause abscess in other organs, or it may form a part of pyemia. Staphylococcal septicemia are rarely seen. Staphylococcus food poisoning. It is result when food contaminated with the enterotoxin produced by the Staphylococcus is consumed. Example: meat, fish, milk, and milk products. Diarrhea and vomiting occur within 6 hours of consuming the contaminated food. Some cases, the post-antibiotic diarrhea are caused by the enterotoxin. Exfoliative disease. Stripping or peeling of the superficial layers of the skin from the underlying tissue by the action of exfoliative toxin is the pathogenic mechanism in various exfoliative syndromes. Means nothing but we will see mainly the stripping of peeling of the superficial layers of the skin. Like examples we can see bullous impetigo, pemphigus, neonatal, Ritter's disease, toxic epidermal necrolysis. SSS is nothing but Staphylococcus scalped skin syndrome. We will see the diseases of bullous impetigo. This is the bullous impetigo image. This is the image of Staphylococcus scalped skin syndrome. And we will go to toxic shock syndrome, TSS. Multisystemic illness, high fever, hypotension, vomiting, diarrhea. In the severe cases, it may lead to acute renal failure, DIC, and peripheral gangrene and death seen in the females. TSS is produced by the exotoxin F mainly. Lab diagnosis. Specimen collected depending on the lesion, like smear examination, culture. Smear examination shows gram-positive cocci in the cultures, and we will use the culture like blood agar, macronchis, media, selective. Already we had discussed about these cultures and what kind of changes we can see in the cultures also we had discussed. Thank you. Subscribe for more videos.